Chapter 22, Prokaryotes, Domains, Bacteria, and Archaea. This is a petri dish used to grow a bacterial species, Serratia marcescens. It acts as an opportunistic pathogen and that it takes advantage of weakened immune systems. Please know that not all bacteria are harmful to life on Earth. In fact, of the more than 5,000 species of known bacteria and archaea, and again, that's just what we know of, less than 1% of the species cause disease in humans. In the 1960s, there was a scientist by the name of Whitaker that used a five-kingdom classification method. These classification schemes were based on the presence-absence of a nucleus, presence-absence of a cell wall, single or multicellularity, and other characteristics. Those kingdoms included Kingdom Monera, which we consider the prokaryotes now, Kingdom Animalia, Kingdom Fungi, Kingdom Protista, and Kingdom Plantae. With the rise of molecular biological techniques in the 1980s, scientist Carl Woese compared these groups by looking at the small subunit of ribosomal RNA and determined that all life evolved from along three domains. Domain bacteria, consisting of, I bet you can't guess, bacteria. Domain archaea, consisting of the rest of the prokaryotes. Domain eukarya, consisting of organisms with eukaryotic cells, animals, plants, fungi, and protists. Both prokaryotic domains are among the oldest organisms on Earth, having existed for 3.5 to 3.8 billion years. These groups are also ubiquitous, meaning they are found everywhere, and I mean everywhere, from kitchen surfaces, forest floors, Antarctica, geysers, deep sea vents, and even areas with no oxygen. Prokaryotes mostly get recognition for the diseases they cause or their use in food production, such as many dairy products. But as mentioned earlier, less than 1% of species actually harm humans. In fact, eukaryotic life would not survive without processes carried out by prokaryotic life. Prokaryotes supply oxygen and produce natural fertilizers taken up by plants and then animals. They are small, but their significance can't be matched. Prokaryotes are everywhere. In fact, your body alone harbors around 1,000 species, mainly because your body provides a nice, warm, moist, nutrient-rich environment. In fact, your own body is an ecosystem, which is just a series of communities of organisms interacting with each other alongside many abiotic or non-living factors. The Earth and Moon first formed about 4.54 billion years ago. For the first 500 million to 1 billion years ago, Earth was devoid of all life. In fact, very little oxygen existed. The temperatures were hot, and the radiation from the sun was much stronger. Most likely, the first life forms were prokaryotes that could survive extreme conditions, and to this day, they are still examples of these extreme microorganisms. It is thought that deep-sea oceans and areas below the surface of the Earth had the right conditions to form life. In fact, the earliest fossil evidence of microbial life comes in the form of microbial mats, which are large biofilms of bacterial colonies compounding on each other. These can be found in creek beds, in your sink, and many other warm and moist conditions. A good example of a biofilm is the plaque which builds on your teeth. Your mouth is warm and moist, contains foods that stick to your teeth. As a result, plaque builds up with bacteria eating away at both the food and your teeth. So brush and floss, people. Microbial mats or biofilms can be quite complex, being several layers thick. They can also be quite colorful based on the metabolic capabilities of the bacteria involved. Here's an example of a microbial mat right here. The first life forms likely got their energy from chemicals near hydrothermal vents, which is a fissure in the Earth's crust and oceans that release heat. Heat speeds up metabolic behavior, and any chemicals that dissolve in the early ocean water will be used as an energy source. Eventually, some groups of bacteria began undergoing photosynthesis around 3 billion years ago and used sunlight as energy source instead. So for the first 2 billion years of Earth's existence, it was devoid of molecular gaseous oxygen. Because of this, only anaerobic organisms, that is, those that grow without oxygen, survived. As time passed and more prokaryotes underwent photosynthesis, a special group of cyanobacteria produced oxygen as a waste product. The rise in cyanobacteria released more atmospheric oxygen and ozone. Therefore, it would be possible for larger eukaryotic life forms to survive and be shielded by the damaging rays of sunlight with ozone in the atmosphere. And just to clarify, ozone is technically O3. That's ozone. Since early prokaryotes could survive in anaerobic, high temperature, high salinity, and extreme pH conditions today, it's likely that they existed in early stages of life on Earth. Even if prokaryotes can't survive these conditions, they may have cell walls, and some can become endospores, which are dormant versions of prokaryotic cells. Regardless, these adaptations allow these groups of organisms to become ubiquitous. So while some prokaryotes live in normal habitats like the soil, fallen logs, your refrigerator, and your gut, some prokaryotes thrive in extreme conditions. For that, we give them the title of extremophile. Here's a list of extremophiles and their preferred conditions. You have acidophiles, which prefer a pH of 3 or below, alkalophiles, which prefer more alkaline conditions of a pH 9 or above, thermophiles like really hot temperatures, and hyperthermophiles like even really hot temperatures up to and including boiling. Psychrophiles prefer freezing temperatures. Halophiles really like high salt conditions like that of the Dead Sea in Salt Lake City around Utah. And then osmophiles like to live in high sugar concentration environments. Studying bacteria is no easy feat. Fortunately, disease-causing bacteria aren't as difficult to extract, grow, and study as these conditions can be replicated in a lab. And anyway, there is a theory of how disease is transmitted. It's called the germ theory of disease, which says that infectious diseases are caused by bacteria and viruses. But you could also include fungi and certain protists in there as well. Typically, a sample is taken from a surface or body part and it is transferred into a sterile petri dish or broth solution. It is then placed in an incubator where it provides a stable temperature for growth and then examined after. Typically takes 2 to 4 days, and after you open up the incubator, you end up with what we call a culture. And each one of those individual areas are called colonies. Much of our understanding of pure culture technique and germ theory of disease is due to a physician named Robert Koch. He developed Koch's postulates, and they read that the microbe must be present in individuals suffering from disease and absent from healthy individuals. The organism must then be isolated and grown in a pure culture away from the host organism. When you look at this petri dish, this is not a pure culture, as a lot of these cultures are starting to overlap with one another and so on. A pure culture would only show one particular species of bacteria, and not just one species, but an entire colony that is composed of nothing but the same actual individual. So in other words, clones. The third postulate is that if an organism from the pure culture is injected into a healthy experimental animal, the disease symptoms should appear. And finally, the organism should be isolated from the diseased experimental animal, again grown in pure culture, and demonstrated to be the same organism. And these postulates have helped us understand how and what causes strep throat, staph infections, gonorrhea, and other many types of diseases that affect humans. So now I want to look at the structure of prokaryotes. So remember, all cells have the following features: a plasma membrane to separate the interior from the exterior of the cell, a cytoplasm which makes up the fluid inside of the cell, DNA which holds the genetic information of the organism, and ribosomes which takes info from DNA to make proteins. The cells fall into 2 major categories: prokaryotic cells or eukaryotic cells. Prokaryotic cells make up single-celled organisms like bacteria and archaea. Eukaryotic cells make up organisms like plants, animals, fungi, and protists. So this table displays the major differences between both groups. Organelles, or membrane-bound compartments, are absent in prokaryotes, while eukaryotes contain several. In prokaryotes, DNA is circular and found in a region called the nucleoid, which is not readily defined. In eukaryotes, DNA is linear and located in a membrane-bound nucleus. With very few exceptions, prokaryotes are much smaller than eukaryotes, and to date, all prokaryotes discovered are single-celled organisms. Eukaryotes can be, on average, 10 times bigger than prokaryotes, and while single-celled eukaryotes exist, most eukaryotic organisms are multi-celled. Let's focus on the prokaryotic cells for this chapter. These were likely the first cells found on planet Earth. First, the nucleoid is a region that holds the genetic material of these organisms. However, it seems to just float inside of the cell. In addition to the nucleoid, prokaryotes can have plasmids. Plasmids, which are separate strands of circular DNA. Outside of the plasma membrane are a couple of layers of protection. First, the cell wall, which also provides shape to the prokaryote, and many prokaryotes have a protective capsule which protects prokaryotes from dehydration and provides more reinforcement to the prokaryote shape. This cell wall is made up of peptidoglycan. Then the flagella, which is used for locomotion, or a pili, which could be used to help transfer genetic information between prokaryotes in a process called conjugation. So as mentioned earlier, there are two domains of prokaryotes, bacteria and archaea. And so while all cells contain membranes, the composition varies slightly between the domains. Prokaryotic cell membranes consist of a thin lipid bilayer made of phospholipids that are semi-permeable. Bacteria and eukarya membranes are made of unbranched fatty acids with an ester linkage, which is just a type of carbon grouping. Archaea membranes are made of a branched fatty acid chains with an ether linkage. In addition, some archaea have phospholipid bilayers that fuse into one monolayer, as shown here. Cell walls do not exist in all eukaryotes, but if they do, they are made up of cellulose or chitin. We'll discuss those in another chapter. In prokaryotes, almost all cells have a protective cell wall. The cell wall provides shape and rigidity to the high osmotic pressure inside. Bacterial cell walls contain peptidoglycan, which is a polysaccharide that contains peptide bonds to hold it together. Due to this, bacteria can be split into two major pseudo-classification groups, gram-positive and gram-negative. This is named for the Gram staining method made by Danish scientist Hans Christian Gram. This four-step procedure includes differing reagents that will color the bacteria purple if gram-positive and pink if gram-negative. These color differences are due to the chemical makeup of each cell wall type. The chemical makeup is due to the amount of peptidoglycan in each group. Gram-positive bacteria have a thick peptidoglycan cell wall. Evolutionarily, these bacteria likely share a common ancestor and form a monophyletic clade. Gram-negative bacteria have a thin peptidoglycan cell wall with an additional lipid bilayer on top. This group encompasses many bacterial clades. Archaea cell walls do not have peptidoglycan, but have pseudopeptidoglycan, which contains a different set of sugars than peptidoglycan. So here's some other similarities and differences between bacteria and archaea. Bacteria do not undergo mitosis due to having no nucleus. The nucleoid, which is shown in red, copies itself and distributes itself to opposite sides of the bacterium. It then develops a septum to divide the cells, allowing the cells to break off of one another. However, the key to life is genetic diversity. More diversity of genes amongst the population increases the chances of a species to survive changing environmental conditions. So how do asexual prokaryotes achieve this genetic diversity? Well, since sexual reproduction doesn't exist in prokaryotes, meiosis can't happen. So how do we get genetic recombination to occur to help increase genetic diversity? Remember horizontal gene transfer from the evolution chapter? The three most common ways in which prokaryotes undergo horizontal gene transfer is transformation, in which they just happen to take up a piece of free DNA and its surroundings. But remember, conditions have to be ideal for this to occur, as genetic material is very fragile. Transduction can occur when a virus seems to intermittently take some harmless DNA from another bacteria, infect that host bacterium, and put that particular bacteria, that genetic material, into the bacterium's own genome. And then there's conjugation. And this happens when one prokaryote forms a hollow tube called a pilus, and this pilus is used to transfer genetic material to another species. Up next, we're going to be looking at the different metabolic components and capabilities, as well as the diseases caused by prokaryotes.